Bueno, buenas tardes. Gracias por su paciencia. Realmente me da muchísimo gusto presentar a la doctora Walderez Dutra de Brasil. La doctora estudió una licenciatura en bioquímica e inmunología en la Universidad Federal de Minas Gerais en Belo Horizonte, en Brasil. Su doctorado en inmunología y bioquímica también fue de la Universidad de Minas Gerais. Después, la doctora realizó una estancia en DINAX Research Institute en California y una estancia postdoctoral también en la Fiocruz Osvaldo Cruz Foundation de Belo Horizonte. Y del 97 al 2008 fue profesor asociado en la Universidad de Minas Gerais en el Departamento de Morfología de Belo Horizonte. Y del 2008 a la fecha es full profesor también en la misma universidad en el Departamento de Morfología. Ha sido profesor visitante en la Universidad de Cambridge del 2018 al 2019 y director general del programa de salud en la UFMG también en el Millennium Park en California. La doctora ha recibido diferentes reconocimientos. Es ahora la futura presidenta de la Asociación Latinoamericana de Inmunología próximamente, actualmente secretaria. Ha participado también en diferentes sociedades, en la Sociedad de Inmunología de Brasil, y ha hecho contribuciones muy importantes al entendimiento de la inmunopatología de la enfermedad de Chagas. Publicado su trabajo en revistas importantes en el área de inmunología, y realmente es un placer que hayas aceptado la invitación y que hayas estado ayer con los estudiantes y esta tarde con nosotros. Muchas gracias, Walderez. Y hoy nos va a presentar How to Mend a Broken Heart.
Sí. So thank you very much, first to Yvonne for remembering my name, indicating my name for this invitation, and then to Gustavo for bringing me here. It's been a pleasure to be here to meet you all. We had a really wonderful time yesterday in our 3-hour talk and then our coffee downtown. So thank you very much to the host team especially, and to all the students in the class. It's really a pleasure to be back in Mexico. This is my second time in your beautiful country. And I truly hope that this is only the second of many times here. And of course, as I have already said to you, to all of you that wish to come visit us at the Federal University of Minas Gerais in Brazil, it will be a pleasure to host you there. So this is where we are. This is so you know the distance that separates us. This is my region in the state of Minas Gerais in Brazil. Next slide, please. And this is pictures of our state. As you can see, it resembles a lot of places that you see here in Mexico. We have many similarities between our countries. We also have beautiful waterfalls and lakes, as you have here the cenotes. And so this is pretty much the pictures of our state. The next slide, please. Not only we have good places to go, but I have to say that we have the best food in Brazil. So I hope that by now I have convinced you to come visit us in Belo Horizonte. If I have the next slide, please. And this is the university. So this is the main building. Unfortunately, the image is not so good here, but you can see that this is the main building of Federal University of Minas Gerais. This is the Institute of Biological Sciences, and this is located in this region here, near the Lake of Pampulha in Belo Horizonte. So that's where the university is located. Our university, I thought it was a very big university, the second biggest university in Brazil, until I learned that UNAM has 300,000 students. We have 50,000 students, so we are very chiquitos compared to UNAM, but the second largest in Brazil. The next slide, please. So today, I want to continue the discussion, but also present to those of you who were not in the meeting yesterday some of the research that we've been doing in our lab in Brazil, dealing with Chagas disease. But the big question here is, how can you mend a broken heart? I guess this is a question that so many of us have asked many times, and we really wish we could answer that question more easily. But let's see about that. Next one, please. Next one. Yeah, so this is a question, right? How can you mend a broken heart? Obviously, the first thing that we should think about when we want to avoid that is to avoid the heart to be broken in the first place. But as we are alive, we are exposed to conditions that may break our hearts, so it's very difficult. We have no preventive measures, and the exposure to events that may break our hearts is inevitable. So this is something that is very difficult to do. However, once you are exposed and affected to the agent that breaks your heart, whatever that agent is, what should you do? Obviously, the first thing is to get rid of that agent. Yeah, get rid of it. But sometimes that is much easier said than done. Not always we can get rid of something that hurts us. And the other thing, which is much more inside of our control, is to control the intensity with which you react to the agent that's trying to break your heart. And that is what our approach has been, working with the disease that I'm going to present to you guys today. If I can have the next slide, please. And this disease is Chagas disease, and I'd like to remind you all that Chagas disease is caused by the infection with the protozoa Trypanosoma cruzi. And one of the main issues with Chagas disease is because it causes a very bad, a very severe heart disease. So Chagas disease evolves from an acute phase that is characterized by a very high parasitemia in the blood and also in other affected organs, and it lasts between 1 and 2 months. During this period, the immune system does a really excellent job at controlling the parasite. So we go from a very high parasitemia in the blood to a very low parasitemia in the blood, and that is when the individuals enter the chronic phase of the disease. The reason why this progression happens most of the time is because it's very hard to really identify Chagas disease clinically, because it gets confused with many other diseases. And remember that this disease is a disease that happens a lot in remote areas. In Brazil, the infection happens in remote areas, so it's hard to have access to hospitals. So the disease progresses to the chronic phase, in which, although parasitemia is very low, this is where main bad symptoms happen with the patients. However, in the chronic phase, we have the great majority of the individuals, around 70% of the individuals, remain in a form that we call the indeterminate clinical form. In the indeterminate clinical form of Chagas disease, patients have no clinical disease whatsoever, no clinical symptoms. We only know that patients have Chagas disease because they have a positive serology. But about 30% of the individuals will develop a very severe cardiac disease that can lead to death of many of these patients. And this death is caused typically by the enlargement of the heart, which is a result of an inflammatory reaction. So indeterminate individuals have no inflammation, or at least no patent inflammation that leads to symptoms, whereas cardiac patients develop a very severe inflammation in the heart that can lead to a very severe disease. So the big question here is, what drives this development from an indeterminate clinical form to the cardiac clinical form? And obviously, what can we do to avoid this transition? So ideally, of course, none of us want to get Chagas disease, but if you do, ideally you would want to remain in the indeterminate clinical form. And so we want to avoid developing the cardiac disease. So talking about how can we mend a broken heart, obviously, the issue of avoiding it in Chagas disease is impossible, because we have no vaccines available currently to deal with Chagas disease. And to control it, we do have a specific chemotherapy, but this chemotherapy is efficient in the acute phase, but not as efficient in the chronic phase, although it still has some efficiency. And the issue with it is that it's very It's very toxic, and not all patients will tolerate this. So the idea here is to come up with strategies that will help control this inflammation, to try to avoid and to try at least either to fix this broken heart or to avoid it getting broken in the first place. And that's what we've been working in our lab, and I'd like to share with you some data that helps us to try to modulate this inflammatory response and perhaps avoid the establishment of cardiac disease or the progression of the cardiomyopathy. The next slide, please. Okay, so Chagas cardiomyopathy, as I mentioned, is a very inflammatory cardiomyopathy. In this graph here, you see in the blue line the measurement for different cytokines for patients with Chagas disease. And in these lines here in the middle, we have different cardiomyopathies: idiopathic cardiomyopathy, rheumatic heart disease, and non-Chagas individuals. And as you can see, there is a general production of inflammatory as well as anti-inflammatory cytokines in Chagas cardiac patients. This is also translated in the number of interactions that these soluble factors display when you look at Chagas cardiomyopathy compared to an idiopathic cardiomyopathy. So clinically, they're very similar, but as you can see, the immune response is completely different. And the Chagas cardiomyopathy not only leads to the production of a lot of inflammatory and anti-inflammatory immunological factors, but also leads to a high number of intense interactions between different proteins in the body. So it's a very intense inflammation that can lead to this cardiomyopathy, and that is why it is not trivial to try to regulate it. The next slide, please. So this slide summarizes to us the importance of having a balanced immune response, obviously not only in Chagas disease, but specifically in Chagas disease in this case. And I remind you that Trypanosoma cruzi, although it is a very promiscuous parasite that infects many different cell types, it likes to live inside of immune cells that are able to present antigens. These cells, called antigen-presenting cells, can harbor Trypanosoma cruzi, and it will present antigens to cells from the adaptive immune response, T cells, CD4 positive T cells, CD8 T cells, double negative cells, and will trigger an immune response. This initial immune response, that is an activating immune response that happens upon infection with Trypanosoma cruzi, is very important. So at this moment of the infection, we do want an inflammatory response. We do want an activating response with the production of TNF-alpha, with production of gamma interferon and other inflammatory cytokines. And the reason for that is because this inflammatory environment will activate the cells that have the parasite inside, and it will help the cells kill the parasite, get rid of the parasite. However, if we don't control this response, this activation can lead to tissue destruction, because a lot of inflammation will lead to tissue destruction eventually. That is why it is important that this side of the equation comes into term with the production of anti-inflammatory cytokines such as IL-10. So IL-10 is an important cytokine because it will decrease the activation of the cells that activate the further immune response to control the parasite and to control the infection, and this will avoid the tissue destruction. As you can imagine, if I control it too much, these cells, the parasite will start growing back inside of the cells. So we sort of need a balance between the inflammatory and the anti-inflammatory environment. So what we've been studying in our lab is not only the production of different factors and the association of these different factors with the different clinical forms of Chagas disease, but also which cells are responsible for the production of these different inflammatory and anti-inflammatory cytokines. And the idea behind that is because if you completely block a particular inflammatory cytokine, you may obliterate the ability of the host to control the parasite. So what we want is to find a specific cell type that I can block without interfering with the other cell types in such a way that I can decrease the inflammatory reaction, but not decrease the ability to keep the control of the parasite. So I would say that we want to use this type of approach, this type of studies, to apply this knowledge generated in these studies of immunoregulation to drive new therapeutic interventions, and our approach is that one of trying to control the intensity with which we react to the infection to Trypanosoma cruzi. The next slide, please. Okay, so this slide here presents the expression of different inflammatory and anti-inflammatory cytokines to make the point for you that inflammatory cytokines such as TNF-alpha, IL-6, gamma interferon are more expressed by patients with the cardiac clinical form of Chagas disease, which makes sense because we know that these patients have an inflammation in the heart, yes? On the other hand, cytokines such as IL-10 and such as IL-17 are more produced by cells from individuals of the indeterminate clinical form. That is the more controlled clinical form of Chagas disease, and these patients, as you know, don't have any cardiomyopathy, so that also makes sense. Now, if I take the cytokines and plot the expression of them along with many other factors, such as chemokines and other soluble factors that you find in the plasma of these patients, as you can see here, you can have a separation of almost perfect between cardiac and indeterminate patients, cardiac in red, indeterminate patients in green. These are the non-Chagas patients, and you can see the association, for example, of the expression of inflammatory cytokines more elevated in individuals with the cardiac clinical form. This can also be observed here in this principal component analysis, in which you have cardiac patients separated from indeterminate individuals, demonstrating to us that they do have a very different immune response. But look at these interesting patients here. Here in the middle, we have patients that belong to the indeterminate clinical form, invading the profile of the individuals that have the cardiac clinical form. And the interesting thing is that 60% of these patients that sort of looked like the patients of the cardiac clinical form, that can be found here as well, 60% of them evolved to the cardiac clinical form after a 3-year follow-up. So that is a result that made us pretty happy, because it's one possibility that we can, by measuring the cytokines in the plasma of indeterminate individuals, if we find a signature that looks like this, perhaps we can predict which patients will move to the cardiac clinical form. And this here is just to show you that these inflammatory cytokines that are more present in the patients with the cardiac clinical form are correlated with a cardiac function that is the left ventricular ejection fraction, which is the ability of your heart to pump blood. So the higher your left ventricular ejection fraction, the better your cardiac function. And as you can see, the more inflammatory cytokines you have, the lower your ejection fraction, meaning that, yes, this inflammation is also relevant clinically for these patients. And the opposite can be seen with IL-10 and IL-17, which, as you know, is more frequent in patients with the indeterminate clinical form. Next one, please. Okay, so all the studies that we do in the lab, most of the studies that we do in the lab, we do with peripheral blood from Chagas patients for obvious reasons, right? The target organ being the heart makes it very difficult for us to have access to biopsies of these hearts. And we end up having access to hearts that have patients that undergo transplantation and all that, which is a good source of information, but only deals with people that are at the terminal point of the cardiac disease. So we do studies in peripheral blood because then we can study individuals with the indeterminate clinical form, mild cardiomyopathy, and severe cardiomyopathy. And the question always is, how does the blood represent what's happening in the heart? So this is the question that we get from many of the reviewers from our papers. And this result here made us very happy, because what we did is we looked at the plasma expression of those different factors that I mentioned to you in the previous slide, inflammatory and anti-inflammatory cytokines, as well as chemokines. And the pattern that we observed here with increase or decrease in particular elements in the peripheral blood of the patients had a 75% correspondence when we looked at the gene expression of the same factors in the heart. So it's very clear that it's not absolutely identical, but we have a 75% concurrence, at least of the immunological factors that we evaluated in the blood and the heart of patients with Chagas disease. So this is important because it means that the blood is indeed an important source of information as for the immune response of these individuals. Next one, please. Okay, so we knew that the inflammatory cytokines were associated with the cardiac clinical forms, and we knew that the anti-inflammatory cytokine IL-10, as well as the modulatory cytokine IL-17 in the case of Chagas disease, is associated with the determinate clinical form. The reason why I have this be careful in calling IL-17 modulatory is because, as we know, it is an inflammatory cytokine associated with autoimmunity. But in the case of Chagas disease, to counteract our hypothesis when we studied IL-17, we thought for sure that it was going to be increased in patients with cardiac disease, but it was actually increased in patients with the indeterminate form of Chagas disease. And then later on, we learned that it's because this cytokine helps in killing the parasite by different means. But in this context, I will call IL-17 as a regulatory modulatory cytokine. But remember that if anybody asks you if Val said that IL-17 is a modulatory cytokine, she said yes, but only in the case of Chagas disease, yes? Otherwise, Yvonne, your immunology teacher, may fail you. So don't do that. Okay, so we knew that there was an association of these inflammatory cytokines with the cardiac clinical form and the regulatory cytokines with the indeterminate clinical form. And then came the question of who were the cells that are important for the production of the cytokines. Okay, so Chagas disease is a disease that lasts for the entire life of the patient. 20 years, 30 years, many, many years. Therefore, the chronic immune response, the adaptive immune response is very, very important. So when we were looking at the production of cytokines in the lab, we tended to look at the response by CD4 cells, by CD8 cells, also by macrophages, but within the adaptive response, we mainly focused on CD4 and CD8 T cells. We published some papers on that. But then I had a really bright student come into the lab. And one day he did an analysis that, just came from his own curiosity, which pointed us into a very, very interesting direction. What did he do? He took the amount of gamma interferon that was produced by cardiac and indeterminate patients as an inflammatory cytokine, and IL-10 also produced by indeterminate and cardiac Chagas patients, and he determined what was the contribution of CD4 and CD8 cells to the production of those cytokines. And look what he discovered. He discovered that only 45% of the interferon gamma that is predominant in cardiac patients came from either CD4 or CD8 T cells. And he also discovered that only 27% of the IL-10 that is predominant in the indeterminate clinical form came from either CD4 or CD8 T cells. So that led us to think that since CD4 and CD8s were not producing most of the cytokines, who was producing it? So we went to look at this cell population that we had not yet studied in the lab until then, that are the double negative T cells. If I can have the next slide, please. The double negative T cells turned out to be very interesting for us to study in Chagas disease. And these cells, although they are at a very low frequency in the peripheral blood of humans, they can express alpha beta or gamma delta T cell receptor that can recognize antigens. And this antigen recognition is done by the presentation through a specific type of antigen-presenting molecules that belong to the CD1 family. So these cells recognize antigens via the CD1 family, and the CD1 family presents mostly glycoconjugates. And why is that so important in Chagas disease? It is because the surface of Trypanosoma cruzi is decorated with a huge number of glycoconjugates, of glycoproteins and glycolipids that will be preferentially presented by the CD1 molecules and therefore will preferentially activate this particular type of cells. And we had the indirect indication of that because of Paulo's analysis of the cytokine expression. The other thing about this CD4, these double negative T cells, is that because they lack the CD4 and the CD8 co-receptors, they can tolerate chronic stimulation. And this is very important in Chagas disease because, as I mentioned to you, it lasts for many, many years. So this really seemed like a very important type of cell for us to study. So we went on to study the cells in the lab. These papers have been published showing that they indeed make a lot of cytokines in Chagas disease. And the interesting thing here is, although they can produce both inflammatory and anti-inflammatory cytokines, if you do a ratio of IL-10 to gamma interferon, look what happens. It will always be toward an inflammatory profile in the cardiac patients, and it will always be toward a balanced profile in the indeterminate individual. So these cells really seemed like a very important for controlling the inflammatory cytokines in Chagas disease. The next slide, please. So all these studies, if I can have the next slide, please. So all these studies that we did to determine the production of cytokines by the double negative T cells, like I mentioned to you, were done with cells from the peripheral blood of chronic Chagas patients. That means that we are studying memory cells in these patients because they have a long-lasting disease, yes? So this is just to remind you, this is from a paper, a review that we published a few years ago, but it comes from the experiments done by Antonio Lazavecchia, describing the formation of our memory cell populations, showing that we can have naive cells be transformed into effector memory cells or central memory cells. These two types of memory cells that I call your attention to are important because the effector memory cells that appear from the activation of naive cells, they can go directly to the site of infection, directly to the site of injury, and perform your effector functions, as the name says. The central memory cells are very important because these are the cells that will be circulating in your body. And if you get in touch with that infection again, it will be activated in the lymph nodes. It needs to go by your lymph nodes, and then it will migrate to the site of infection and fight your infection. So having these two populations is very, very important. And as we will see, they may have completely different functions and activities in several diseases. Until then, only CD4 and CD8 populations had been studied in Chagas disease by Albareda in Argentina and by Fiuza in Brazil. And we then centered our attention into defining the memory profile of those double negative T cells that we thought were really the most important producers of inflammatory and anti-inflammatory cytokines. If I can have the next slide, please. Okay, so how do we study memory cells? How can we do that in vitro? So what we did is we took cells from patients of the indeterminate and the cardiac clinical form of Chagas disease, and we performed flow cytometry with these cells, first to identify the double negative T cells. We use CD4 and CD8 markers in the same axis here. We selected the cells that were negative for CD4 and CD8, but that did express the TCRs, either alpha, beta, or gamma delta. So after selecting these double negative populations that we knew were T cells because they had the T cell receptor, we utilized markers that can define the different populations of memory cells, particularly central memory and effector memory cells. So these two markers, CCR7 and CD45RA, are glycoproteins that are expressed differently in the membrane of the cells that can characterize these different cell populations of central and effector memory, according to Salusto and collaborators. So we did that, and we centered our attention exactly in the central memory and the effector memory cells, thinking of the effector cells and the ones that keep your protection in your bloodstream. The next slide, please. Okay, so here they are. So when we first looked at this type of analysis, that is the TSNE analysis that I actually mentioned to you guys yesterday, which is the T-stochastic neighbor embedding analysis that takes many markers and distributes these markers in relation to each other, plotting them in different positions as you see here. the more red you have, the more of that marker you have, the less red you have, and more blue, the less of that marker you have. So if we take these two markers, CD45RA and CCR7, and plot them using the t-SNE, what we can identify here is the central memory population. Here we have the effector memory population that I call your attention to. So central memory up here, effector memory up here. If I now take this plot and separate my patients into the cardiac and the indeterminate clinical form, it is very clear that the cardiac patients have a predominance of this population down here, much more than this one here, which is the effector memory population, whereas the indeterminate patients have a predominance of this population down up here, which is the central memory population. So right here we see a big difference between the populations of double negative T cells in the Chagas patients, in which the double negatives that I see in cardiac patients are mostly effector. The double negatives that I see in indeterminate patients are mostly central memory cells. If I can have the next one, please. But then you can quantify that as well. You can take the numbers from those plots that I showed to you. And here in these plots, we can see that, in fact, if I look at double negatives expressed in either the alpha beta or the gamma delta T cell receptor, I can see that there is a predominance in central memory population in indeterminate as compared to cardiac Chagas patients. And I can see that I have a predominance of effector memory, as we saw in the plots before, in the patients with the cardiac clinical form of Chagas disease. If I can have the next slide, please, and the next. Thank you. So now we know that the central memory is predominant in the indeterminate patients. We know that the effector memory are predominant in the cardiac patients. But if I take this now and look at that very same indicator of cardiac function and plot it against the frequency of central memory or effector memory cells, look what we observe. We find that the higher the frequency of central memory cells, either alpha beta or gamma delta, but predominantly gamma delta, we see that we have a better cardiac function. And the opposite is seen when we look at the effector memory population. So the higher the frequency of effector memory population, the worse the cardiac function. So that all made sense, not only regarding the inflammation, but also regarding the clinical characteristics of this patient, showing that this population did have an association that seemed to be important. If I can have the next one, please. Thank you. Okay, so we had shown that these cells were able to produce both inflammatory as well as anti-inflammatory cytokines, as I mentioned to you. This had been previously published, and it was very interesting because they really can make all kinds of different cytokines. However, Dragana Jankovic in 2007 had shown a very particular cell population that produced concomitantly IL-10 and gamma interferon. This cell population was described in patients that had toxoplasma infection, and this population was clearly associated with a protective response. The reason that Dragana gave for that is that these cells, although they can produce gamma interferon, which is highly inflammatory, they also have the ability to control themselves because they produce IL-10 themselves. So we went on to look at the production of both cytokines by these populations of central memory and effector memory in indeterminate and cardiac Chagas patients. And what we saw is that the central memory population in the indeterminate patients, as well as the effector memory population, had a much greater production of both IL-10 and gamma interferon at the same time, suggesting that, in fact, the central memory population was again associated with a more protective response. The next one, please. Then we went on to look at the activation state of these cells, of these different memory populations. And for doing that, we evaluated the expression of CD69, which is a marker of T cell activation. And we found a very interesting thing, and I'll call your attention to the fact that the central memory cells from the patients with the cardiac clinical form, whether they were alpha beta or gamma delta double negative T cells, when you stimulate the cells with triple mastigold antigens, they decrease the frequency of cells expressing CD69 instead of increasing upon stimulation, they decrease the frequency of the cells expressing CD69. When we look at a plot, plotting CD69 and the protective cytokine IL-10, look what happens. The cells from the indeterminate patients that are activated are mainly producing IL-10, and this falls into the area that is occupied by the central memory population. However, the cells from the cardiac patients that lose the expression of CD69, that are not getting activated, are not producing IL-10. So, and you can see that quantified down here, that the production of IL-10 by activated cells is much lower in patients with cardiac disease. So these patients are lacking the central memory population that produces the anti-inflammatory cytokine IL-10, and that may be contributing to their disease and to that cardiac disease. So the question is, so the next one, please. So the question is, could we rescue this balance? Could we do something to prevent these cardiac patients, the central memory cells from the cardiac patients, to becoming so inflammatory? How could we do that? So I had also a very excellent student in the lab, Livia Passos, and Livia suggested that we blocked the activation of double negative T cells by blocking the interaction with that CD1 molecule. Remember that CD1 molecule is the one that presents the antigen to the double negative T cells. So she had the idea of using an antibody that would bind here and prevent disactivation of the double negative T cells. And I put the CD1D molecule here, although we know that the CD1 family has other molecules, because she had done previous studies and showing that CD1A, B, and C were not as important as CD1D for presenting the antigens, and that's been published in JID a few years ago. So based on that, we decided to block this molecule, this interaction here. If I can have the next, please. And the idea behind that, oh, this is just to show that CD1D was the most important. So the idea behind that is that if we block the activation of this memory population in cardiac patients that is not producing IL-10, perhaps I can rescue a better response. The next one, please. So what she did is she took cells now only from patients with the cardiac clinical form of Chagas disease. She exposed the cells to triple mastigote antigen in the presence of that blocking anti-CD1D antibody, and then we evaluated the expression of inflammatory and anti-inflammatory cytokines. If I can have the next, please. But first, what she did was to look at the activation of the cells. So the idea was to decrease the activation of the double negative T cells. And here she actually showed that we were actually able to decrease the activation of the double negative T cells of beta, but mainly the gamma delta subpopulation. So we lowered the activation of the cells here. And importantly, she did not see a decrease in activation of CD4 or CD8 T cells. That was expected because we blocked the CD1D interaction, but it's important to maintain the activation of CD4 and CD8 T cells because the cells need to be active to be help controlling the parasite, right? So that was good that you only tempered with the activation of the double negative T cells. The next one. So look what happened here when she looked at the expression of central memory and effector memory populations. Remember that the effector memory populations is the predominant population in patients with cardiac clinical form of Chagas disease. When she blocked that activation, she increased the expression of the anti-inflammatory cytokine IL-10, and importantly, she increased the frequency of double negative T cells of the effector memory population that express both gamma interferon and IL-10. So in fact, by blocking the activation of these double negative T cells, she was able to rescue an anti-inflammatory profile in the patients with cardiac Chagas disease, suggesting that tampering with the activation of double negative T cells could be a good strategy for decreasing inflammation without dealing with CD4 and CD8 that would still be active to help control the parasite. And as you see here, this difference was observed in gamma delta, particularly in gamma delta T cells. So we are also, and as you saw in many of the results that I showed before, the gamma delta subpopulation was the one that was more responsive amongst the double negative T cells. So we wanted to see a little bit more about this gamma delta double negative T cells. If I can have the next, please. So who are these gamma delta T cells? We saw that within the double negative subpopulation, these cells were most responsive to the activities that we saw in the memory population of the cardiac patients. But we also know that the gamma delta T cells can be CD4 positive, can be CD8 positive, or can be double negative, as you saw. These gamma delta T cells, they can be stimulated by a number of antigens, but again, here we have the glycoantigens that are predominant in the surface of Trypanosoma cruzi. So obviously we see that they are good candidates for activating these gamma delta T cells. Once these cells are activated by the different activators that can be observed here, whether they are CD4, CD8s, or double negatives, they can display a gamma of different functions, such as cytokine production, cytotoxicity, B cell help, dendritic cell maturation, and also priming of other cell populations, such as alpha beta T cells. And the other important point is that these gamma delta T cells do have the ability to cross-regulate each other. So when you think of a chronic disease that can last for 30, 40 years of the patient's life, this is an important thing, because if you don't have some kind of control of this response, obviously the patient would die much earlier of cardiomyopathy, and that's not what we observe. So we wanted to learn a little bit more of all the gamma delta populations, not only the double negatives, but all the gamma delta populations that were present in the blood of the Chagas patients. So if I can have the next one. Okay, so first thing that we observed, this was done by a undergraduate student in the lab. He started in the lab when he was in the second semester, and he started taking over these studies here. And Pedro observed that the gamma delta CD4 and CD8 cells were increased in patients with Chagas disease as compared to individuals that were not affected by Trypanosoma cruzi, and that was particularly important within the CD8 subpopulation. So CD8 gamma delta cells were increased in patients with Chagas disease. Then when they looked at the gene expression in the heart using a public database for doing that, they also observed that although the alpha beta TCR expression was not different in the heart, there was an upregulation of the gamma delta gene in the heart of the Chagas patients. This was interesting to us because typically these gamma delta cells are found in mucosal tissue for the immune responsive mucosal tissue, but you saw that they were increased in the peripheral blood, and they were also increased in the heart of patients with Chagas disease. So that made it even more interesting to us. So if I can have the next. So the first thing that we did was to go back to our population of patients from Minas Gerais and collect blood from indeterminate and cardiac Chagas patients, stimulate these cells in vitro, and evaluate the expression of several different types of molecules that would indicate to us what would be the functional profile of these cells, such as inflammatory and regulatory cytokines and their receptors, such as cytotoxic molecules, such as receptors that could drive the cells to the heart, as well as try to manipulate their function to see if, again, we could use this population to control inflammation and fix our broken heart. If I can have the next, please. Okay, so first thing was the measurement of the cytokines. So I call your attention here for the expression of the inflammatory cytokine TNF, the anti-inflammatory cytokine IL-10, and down here the ratio of TNF and IL-10. When you look at the gamma delta CD4, CD8, and the double negatives, you can clearly see that CD4s and double negative T cells were excellent producers of cytokines, much more so than the CD8 cells, particularly the inflammatory cytokine TNF, and that this was increased in patients with the cardiac clinical form of Chagas disease as compared to patients with the indeterminate clinical form. When we perform the ratio of inflammatory and anti-inflammatory cytokines, what we observe is that, yes, the cardiac patients displayed a more inflammatory profile as compared to the patients with the indeterminate clinical form. And this was particularly predominant within the CD4 gamma delta and the double negative gamma delta, as opposed to the CD8 cells that had very low expression of this inflammatory profile. However, when we looked at the expression of cytokine receptors, what we observed very clearly here is that the CD8 cells expressing the gamma delta TCR have a lot more expression of the receptor for the inflammatory cytokine TNF as compared to CD4 or the double negative T cells. So that means that suggests to us that the CD4 and double negative are producing the cytokines, the inflammatory cytokines, but that the CD8 cells are the ones that are more prone to respond to this inflammatory environment in patients with the cardiac clinical form. The expression of IL-10 receptor was much lower in the cells, again suggesting that they are prone to respond to the inflammatory environment. And if we perform a ratio of TNF receptor and IL-10 receptor, again, we see that this is much higher in CD8 gamma delta cells from patients of the cardiac clinical form. If I can have the next one. So we became very interested in the CD8 gamma delta cells because they seem to be very responsive to that inflammatory environment. And here we evaluated the expression of granzyme and perforin, which are molecules associated with cytotoxic function. And as expected, these cytotoxic molecules are indeed highly expressed in cells from both cardiac and indeterminate Chagas patients. And this is expected because CD8 cells are mainly associated with cytotoxic function, although the double negative T cells can also express a lot of the cytotoxic molecules, as you can see here. But very interestingly, when we look at the expression of granzyme, the only population that displayed an association with a clinical parameter of heart inflammation were the CD8 gamma delta T cells, in which the higher the expression of this molecule, this granzyme molecule by CD8 gamma delta, the worse the size of the atrium, the left atrium of this patient, suggesting a worse cardiac function. So again, suggesting that the CD8 cells may be associated with a worsening of the inflammatory response in the patients with cardiac disease, because as you see, we did not observe the same thing in patients that had no inflammation. The next one, please. Here we can clearly see that the CD8 gamma delta cells are different from patients with the indeterminate and the cardiac clinical form. You can dissociate these cells in the cardiac versus the indeterminate patients, showing that they are much more inflammatory in the cardiac patients as compared to the indeterminate patients. And this is much more clearly seen after you do a recall response in vitro with triple mastigote antigens. And here you see that also in the cardiac patients, although you have the sort of the same types of interactions with the very same molecules that are found in both clinical forms, the number and the intensity of these interactions is greater in patients with the cardiac clinical form, again suggesting a more active immune response in these patients. If I can have the next one. Then we went on to evaluate the expression of receptors that trigger the cells to go to the heart. Because remember, we are looking at the peripheral blood of these patients, and we also saw an increase in gamma delta gene expression in the heart. So the idea was, can these cells that are activated in the peripheral blood migrate to the heart? So we took receptors that had been shown in experimental models of trypanosoma infection as being important to drive these cells to the heart, and as well as a work done by people in the Imperial College in London showing the co-expression of CCR5, which is important in Chagas disease, and this molecule that is c-Met as a sort of like an address code to send the cells to the heart. And when we looked at the expression of CCR5, we see that all the subpopulations, CD4, double negatives, and the CD8 in cardiac patients express more CCR5 than cells from indeterminate patients. But when we looked at c-Met, the expression of c-Met was a lot higher in cells from cardiac patients, and particularly CD8 cells, as compared to CD4 and CD8 cells. So of course, that the co-expression of these two molecules being more predominant in CD8 cells suggests that they might be prone to be migrating to the heart of the Chagas patients. So if I can have the next one, please. So we went on to look at the expression of these molecules in the heart. Actually, this was an expression of genes in the heart of Chagas patients. Here we have again the increased expression of gamma delta that I showed to you before. And in these plots here, we have a correlation of the gene expression of gamma delta with different molecules. So as you can see, there is a correlation of the gamma delta gene with the expression of CD8 gene, as well as the expression of all those molecules that we showed to be increased by the CD8 gamma delta subpopulation in the peripheral blood of this patient. So that was also observed in the heart. I labeled here in red the TNF-alpha receptor because TNF has been associated with severity of Chagas disease in many experimental models. And we know that TNF is a very important inflammatory cytokine. If I can have the next one. This is a study that we did in another disease that showed again that TNF was indeed very important for controlling inflammation. Actually, the best way to say it is that if you can control TNF-mediated inflammatory responses, you can better an inflammatory response and better a disease. So this is a different disease here, also a parasitic disease called leishmaniasis that is caused by the infection with leishmania. This is a mucosal form of leishmaniasis. This is shown here a very swollen septum, and here a normal septum, because this is one of the clinical manifestations of this disease. And in this case, what we observed in Brazil, working in the endemic area for leishmaniasis, is that a lot of patients were refractory to the regular treatment for leishmania, which is pentoxifilin. So we decided, based on data that we had previously obtained in the lab, showing that TNF was important for mediating inflammatory response to leishmaniasis, as well as we saw in Chagas, we decided to combine the conventional treatment with a chemical that blocks or that controls TNF-mediated inflammatory reaction, which is pentoxifilin. And what we observe here in this curve is a very encouraging result, showing that patients that are treated only with the regular treatment and parasitic treatment for leishmaniasis take about a year to get cured, whereas patients that are treated with the combination of the antiparasitic with pentoxifilin have a decrease of over 200 days in the cure of that lesion. So that's very important, but because that allows for those people to resume their normal lives and become active again. So because we had those results in leishmaniasis, and because we knew that TNF-mediated inflammatory response was also important in Chagas disease, we decided to try in vitro to modulate the inflammatory response of the gamma delta T cells that I just showed you in patients with Chagas disease. If I can have the next one. So what we did is we took cells from patients with Chagas disease. We stimulated in vitro with parasite antigens, and in the presence or the absence of this chemical that reduces TNF-mediated inflammation. And then we looked at the characteristics of these populations, hoping that this was able to modulate their function. So if I can have the next one. And as you can see here, if you remember, the CD4 gamma delta cells and the double negative gamma delta cells were important in producing the cytokines, whereas the CD8 gamma delta cells were the responsive to the cytokines. And as you can see, the treatment with pentoxifilin decreased significantly the expression of TNF by CD4 gamma delta T cells. And although it didn't have a direct interference with the double negative T cells, it did increase the expression of the anti-inflammatory cytokine IL-10 in this subpopulation. So it had different activities in CD4 gamma delta or double negative gamma delta, but in the end, it diminished the expression of an inflammatory cytokine and increased the expression of anti-inflammatory cytokine. In the case of CD8, which are the cells that respond to the inflammatory environment, we observed that there was a decrease in the expression of the TNF receptor, which means that the cells would be less responsive to the inflammatory environment and perhaps have a less destructive function, and also decreased a little bit the expression of cytotoxic molecules, suggesting that indeed also in Chagas disease, perhaps pentoxifilin will have the ability to modulate this response. So if I can have the next one. So in summary, what we saw today was that looking at particularly the double negative T cells, we're able to modulate the profile of the cells by blocking their activation and rendering the effector memory population that is predominant in the cardiac patients to a less inflammatory profile when treated with anti-CD1. And when we looked at specifically at the gamma delta subpopulation, we also observed that we can modulate this inflammatory response now using a different strategy, which is the one to decrease the TNF-mediated cytotoxicity and activation of these cells. So in the case of how can we mend a broken heart, in this case, we hope that by controlling the intensity of inflammation, either by blocking the activation of double negative T cells or blocking the activation of TNF-mediated inflammation, we'll be able to decrease the inflammation and perhaps avoid the progression of this cardiomyopathy. So we are now at the point where we'll intend on translating this data to patients. It's important to mention that both molecules, anti-CD1D and pentoxifilin, are approved for use in humans. So we can try and see whether we can prevent the inflammation in these people. And as you saw, by tampering with these cells, we do not prevent them from controlling from having the activity of other cells that can control the parasite. So the experiment that Pedro is doing this week is to see whether this blocking will really lead to not allowing for the parasite to come back. So he's infecting cells in vitro, blocking the response to see whether we can actually keep the control of the parasite. So that's what I wanted to finish with you today, showing that we have potential immunotherapeutic targets to prevent the control of cardiomyopathy. So although it is difficult to mend a broken heart, as all of you and all of us have known by different experiences, but it's possible to do that. And we hope that we can translate this to the benefit of the populations of people with Chagas disease, in Brazil, as well as in other 21 countries in Latin America that are endemic for Chagas disease. So the next one, please. And last but not least, thanks to the people that have been doing this work. This is Pedro, the undergraduate student that did the majority of these studies. This is Eula Neves, who has been working together with him in the cellular part. Tayane Souza Silva, who does all the bioinformatics and in silico analysis. Lívia Passos and Fernanda, former members of the lab that started these studies, really, and all the other collaborators. I particularly thank Maria do Carmo Pereira Nunes and Silvana Araújo, that are the clinicians responsible for the care of the Chagas patients, and Ken Golub, who really has been a very important collaborator in this, as well as many other studies. And of course, without funding, we cannot do research. So I very much thank the funding agencies that have been funding our studies for many years. And with that, I open for comments and questions, and again, invite you all to come to visit us in Brazil at the Federal University of Minas Gerais. Gustavo has my contact, you have my email, so if you want to come and visit and spend some time with us and do some lab work with us, it will be a pleasure to have you there. Thank you.
Iniciamos la sesión de preguntas. Las personas que están en Zoom o en YouTube pueden escribir sus preguntas en el chat y se las leemos acá. ¿Preguntas? ¿Quién se anima?
Thank you, Walderez, for your presentation. It was very interesting. I was wondering if could there be certain antigens in T. cruzi specific that are responsible for different immune responses, and if is it possible to explore the antigen diversity specifically in the antigen presenting cells?
So that's a very important question, and you have a very good point, because the strategies that we used to block the inflammatory response are general, right? So they will block the response in a general manner. Ideally, if we have an antigen, if we identify the antigen, that can activate these cells and use that antigen to mimic or block this activation, that would be ideal, because we would only deal with the cells that are specific for the parasite. So the function of the other cells would keep intact. So that's a very important question. So we did some experiments regarding that. And what we did is we took Trypanosoma cruzi, the triple mass goat form, and we fractionated it in glycolipid and the glycoconjugate fractions that have sugars, lipids, and proteins, only proteins and only lipids. And we saw that the activating part of the parasite was really in the glycoconjugate fraction. After that, we took this glycoconjugate fraction with a collaborator of ours in the University of Texas in El Paso, and he subfractionated that. So now we have a lead that the molecules that are responsible for the activation of the double negative T cells and the gamma delta T cells are within the glycoconjugate fraction and are the ones that are more hydrophobic, because the way he fractionates that is by releasing these molecules from a column with alcohol. So the ones that come out last are the most hydrophobic ones. So we believe that in those two last fractions, there will be the stimulatory antigens for these cells. The idea now is to purify these antigens and see if we can use them to design a molecule that will block the function of the cells. So yes, you just described our R1 grant for the NIH, so that's very good.
¿Alguna otra pregunta?
Thanks. When it comes to Chagas, I was wondering, well, the big question is why the heart, right? Why only we see these big symptoms for the heart? So what I was thinking, and what I was wondering is T. cruzi, the parasite, only found in the heart, or does it also spread to other tissues but only show this harmful immune response when it comes to the heart?
Very good question. So yes, Trypanosoma cruzi also leads to immune response and pathology in other organs. So there is also another clinical form that is called the digestive clinical form, in which the patient has enlarged esophagus or enlarged intestine. So you do have parasites that preferentially go to the GI tract, and you do have an inflammation there. It's a completely different type of inflammation. You saw here that I showed you that association with the heart is TNF, gamma interferon, and in the GI tract, you have a much different inflammation that is sort of like a Th2 type inflammation with lots of cells such as eosinophils, neutrophils. So it's a very different response. But despite being different, it's also very bad. And lots of people die of that as well. Not as many as the cardiac disease, but they also die of that. Regarding other organs, besides the heart and the GI tract, there has been studies that show T. cruzi in the brain. So there is an association with, for example, an increase in brain aneurysm in patients that have T. cruzi infection. And people even have suggested to create a new clinical form called the nervous clinical form, neuronal clinical form, the nervous system clinical form. In Portuguese, I would say the forma nervosa of Chagas disease, but that hasn't really been proved, definitely yet. Other studies have shown that if you treat the patient and if you follow that patient for and this study particularly was 5 years, and you don't find parasites. Remember, I mentioned to you yesterday that you can culture blood for about 6 months and grow the parasite. If you treat the patient and if you follow for 5 years and cannot grow parasites, which means that that patient may be parasitologically cured in the blood, but if that patient gets immune suppressed, sometimes the parasite can come back. And people have suggested that it's because this parasite can be hidden somewhere, but we don't know where. So yes, it can go to other places, but we don't know yet which are all the other places that it can go to. Perhaps you can come to Brazil to try to study that with us.
Thanks.
You see that I'm trying to steal all of your students for you because they're truly very excellent. So yes.
Hi, thank you for your presentation. I was wondering about this last idea about the double negative T cells, making them block the production of cytokines. Would that treatment be would that be like I forgot the word, but would that be have like an effect that is long lasting for all the other T cells, double negative T cells, or the gamma delta?
That's a very good question.
And would it be important, or would that have some negative effect in the immune response later for the patients?
That's two very good questions. The first one is whether how long this activity would last. Yes. So this is something we don't know yet. So we have to translate that into the clinic to be able to determine how long that would last. We know that when the treatment with the anti-CD1D was done for lung diseases, it lasted for about 6 months. We don't know if that would be enough, or how we would deal with that. And that's important because, as you know, most of the people who have Chagas disease need a treatment that is very cheap. We cannot afford giving expensive treatments to people. In that sense, pentoxifilin is a lot better because it's a very cheap treatment, but it has the difficulty of being a general thing. So the side effects, which comes to your second question, could also appear. So we don't know. The last thing we want to do is to decrease the ability of these people to deal with the parasite, to keep the parasite controlled. And if this is one of the side effects, then we cannot use this treatment. So what we have to do is to test that, and also in addition to test whether there will be other side effects that will prevent the people from getting treated. If you're talking about indeterminate patients, and again, we don't know when would be the best time to enter with the therapy. The indeterminate patients don't have inflammation, but they are the ones who will develop the cardiomyopathy. So would it be best to enter with this treatment at the very beginning of the cardiomyopathy, or would it be best to have it established a little bit more and then, so these are things that we need to see. And I was telling the students, were you in the class yesterday? Yeah. So you remember that I mentioned the leishmania treatment. If you give it too early, it's bad. We thought it was going to be wonderful. We educated the patients to go to the clinic because we wanted to treat them right away before the lesion even appeared. And it turned out that it was much worse. So we have to see. Sometimes we create a hypothesis, and when we go do the experiment, the hypothesis is completely different, and it's not what we thought.
Voy a leer una pregunta de Zoom. Itzel Rojas pregunta, bueno, thank you for your presentation. And if there is any genetic factors or another factors associated with the clinical cardiac form of the disease?
Yes. So there has been many studies, including from groups in Mexico and in Brazil and in Argentina, that have shown that there are some genetic factors that are associated with, for example, the establishment of the cardiac clinical form. I'll mention our study that showed that a single nucleotide polymorphism in the promoter region of IL-10 is associated, and that leads to the low production of the cytokine, is associated with the cardiac clinical form of Chagas disease. Another important finding that Tayani, who I showed to you today, just recently had, is so many studies done throughout the world try to associate Chagas with a particular MHC expression, thinking that perhaps, a particular MHC gene would be associated with the development of the disease, and we failed to do that. However, there is a particular motif that is found in different types of MHCs, that this motif is DERAA, 5 amino acids that appear in the HLA-DR 0102, 0104, 1301, and 1302. So these 4 different MHC. So when we look, we try to associate the clinical form with one MHC, failed. Now, if you take just the DERAA, we found that that appears in 60% of the cardiac patients. So perhaps the MHC, after all, has an association that is not directly related to one allele, but rather to a particular motif that you find in the antigen. So Tayane did a really beautiful study that I didn't have the chance to show to you today, but perhaps if Gustavo invites me again, I'll show you, which she took this DERAA and went to do bioinformatics with it to try to find peptides that could be presented by this DERAA. And she identified, very interestingly, she did not find anything from the parasite that could be presented by DERAA, but she found things from the host that could be presented by it. When she took these peptides from the host and then blasted them back into the trypanosome, then she found correlation with proteins from trypanosoma. Remember that I mentioned to you yesterday that there are many proteins from trypanosoma that trigger sort of an autoreactive response. So we went and we synthesized those peptides and we stimulated cells in vitro, and it's very nicely showing that patients who have the DERAA present them really well, induce the expression of inflammatory cytokines, and people that do not have that allele present it a lot less. So it's very interesting association, perhaps with a genetic factor.
¿Alguna otra pregunta?
First, thank you so much for your presentation. And the question is, in a 60-year-old patient who acquired trypanosoma cruzi in her childhood and currently the size of her heart has grown, could this therapy help his heart decide?
So the thing with Chagas is once you establish the cardiomyopathy, you cannot take it back. So the best strategy is really to avoid it progressing to a very bad place, so but once it gets established, you cannot take it back. However, you can help not make it worse. Let's put it that way. So if the person has the cardiomyopathy, you can interfere and avoid that it gets worse, but you will not bring it back to normal activity anymore. That was a long study done by an Argentinian group, by Vioti's group, showing that once you establish it, it will slowly progress. You cannot take it back anymore. But that was before the treatments that can modulate the inflammatory response. So our hope is that by at least modulating that response, we will avoid the progression, but we cannot take it back.
Thank you so much.
Thank you for this wonderful presentation. I would like to go back to what we were talking a few hours ago. And do you know if the people who have, you certainly don't know, but maybe you will look into people who develop the cardiac form of the disease have a higher or lower expression of CD43? As I mentioned, CD43 is a ligand for the T-chromosialidase. So it would be interesting to find whether that has some effect on it. One, because of what it could do for the parasite, for the entrance of the parasite itself, because the APCs clearly also present.
So,
and then also for the kind of response that the patients will develop.
So Yvonne, if I remember now, I think we did use CD43 to classify different B cell populations, if I'm not mistaken. Would it be possible? Is it something that we do? I don't recall. I know that we had CD27, CD19, and I think CD43.
You do use CD43 for B cells.
Okay, so I have not analyzed the expression of CD43 in T cells, but I can do that because I have the FACS files.
Right.
So we published the paper on B cells a couple of years ago in Frontier, showing that the B cells, particularly the B1 B cells from the Chagas patients, are highly activated and producing.
And B1 B cells are highly CD43 positive.
Right. So perhaps that's the marker that we use to select them. So now I can take that data and look at the expression of CD43 in the other lymphocytes.
Oh, yeah.
I hope to be able to tell you that soon. Yes.
Okay.
And maybe in the B1B cells that express the high CD43, the ligand, the sialic acid ligand is the one that is stimulating, huh? That's interesting.
Thank you. Very nice for the very, very, very nice talk. I was wondering if you can correlate the cardiac form of the disease with a previous inflammatory response, for instance, or sex, male, female, or even the age.
Yes, very good question. So yes, the frequency of cardiac disease is higher in male. We don't know yet why. It's interesting because we were doing a study comparing the immune response of patients with rheumatic heart disease and Chagas disease. Rheumatic heart disease is very different because it's a valve disease and it's much more frequent in women. And then we found that the Chagas cardiomyopathy is much worse in males than females, but we don't know why yet. Perhaps there is something to do with hormones or, but we don't have a response to that yet. Age doesn't seem to be related to it because, for example, you can have patients that remain in the indeterminate clinical form until very old age. 85, 90 years old, and they remain at that age. It's also important to remember that the more the time goes by, the higher your chance of progressing to the cardiac disease. So yes, if you wait too long, you will have people that will progress to the cardiac disease. But the age itself doesn't seem to be a factor because you have lots of people that die of other diseases and never have cardiomyopathy remaining in the indeterminate clinical form. So age, no. Sex, yes. And previous inflammation, we don't have information on that. One of the kids, yes, they asked about other infections, parasitic diseases or other things that can affect your response. And that is such an important thing to do in our countries because we are endemic for so many other inflammatory diseases. So for example, dengue. What happens to the patient that has such an inflammation in dengue? Does it worsen the cardiomyopathy? We don't know yet. But, but I need people to come to the lab to do the studies in dengue as well.
Yeah, I think it would be very interesting also to check these gamma delta T cells in other type of infections or diseases, right?
Yes. Yeah. So we studied some of it in Leishmania, and we found that they are also very active in Leishmania. And we are now thinking of looking at it in the bacterial disease because, Trypanosoma and Leishmania are cousins. So it's not too surprising that, but now in rheumatic heart disease, perhaps they have a role as well. And it's funny because since they don't have the co-receptors, we never looked at them. It's only CD4, CD8, and then all of a sudden they show up.
Thank you.
Thank you very much for the lecture. Well, with his question, well, you mentioned that it seems that Trypanosoma cruzi is able to hide somewhere in the body. Is it able to colonize the bone marrow? Because there are certain viruses that are not present in the blood, but when the patient is immunosuppressed, they recolonize the blood coming from the bone marrow.
That's a very, very good question and very important question. So we don't know in humans yet if it can colonize the bone marrow. But there were many studies done by a very brilliant Brazilian scientist, Mauricio Rodrigues, who was trying to develop a vaccine for Chagas disease. And he found antigens that were very protective. So he was able to find protection. But then when he immunosuppressed the animals sometime later, the parasite came back. He had looked for the parasite everywhere that you could think of: brain, heart, GI, skin, muscles, liver, kidney, spleen, everywhere, could not find it. And it turned out that it was in the bone marrow. So in the experimental model, yes, it can stay in the bone marrow. There is another group of studies by Wilson, Savino, and Fiocruz that shows that by living in the bone marrow, it can compromise the development of certain immune cells. Yeah. So because if it gets there at a very early age, remember I told you guys yesterday of the congenital infection, that can influence the repertoire that the individual will have. But these are all done in experimental models. In humans, we don't know yet, but that's a very good question and a very important point, because if that is the case, we need to find things that can reach the bone marrow to be able to get rid of the parasite. It's very difficult to get rid of these parasites because you guys remember, it's been with us for 9,000 years. So our co-evolution is very impressive. You know, 70% of the people have nothing. They just have the parasite and have no disease whatsoever. So, you know, we just go living together, getting better together. The problem is, what happens that you lose that ability to maintain your co-evolution so well? Why are we, and you know, what's going to happen with these other infections that come along with COVID, with things that who knows what? How is this going to interfere with our response toward these agencies that we have to deal on a daily basis that are in our countries, and you know? So this is very important to find out.
Okay, thank you very much.
Thank you.
Presentation.
Pleasure to be here with you all. Thank you very much.